Tulane scientists are working on something that could help our brains and our ability to remember.
What we see is in rats that have a source of estrogen will outperform rats that don't have a source of estrogen on a variety of memory tasks.
Tulane Associate Professor of Psychology and Neuroscience, Dr. Jill Daniel, studies female rats to learn how hormone replacement, specifically the female sex hormone estrogen, for women after menopause can have an anti-aging effect on the brain. And here's what they learned in their study. The rats that got estrogen only in their middle age and the rats that got estrogen from middle age through old age had better memories that lasted longer compared to the rats that got no estrogen.
And they can remember, if they have estrogen on board, up to 5 or 6 hours where the food was hidden in the maze. Whereas if they don't have estrogen, they usually forget after about 30 minutes or so.
The rats studied in this maze have no ovaries, so they produce no estrogen. They get a pellet implant like postmenopausal women to deliver the estrogen. It is clear on imaging of the hippocampus, the part of the brain involved in memory and spatial navigation, that those on hormone replacement have more neurotransmitters and estrogen receptors. In Alzheimer's disease, the hippocampus is one of the first places to show damage, memory problems, and disorientation. Now Dr. Daniel's lab has a $1.4 million grant from the National Institute on Aging to see why short-term estrogen, just given in that critical window right after menopause and middle age, then taken away, continues to protect the brain.
Our rats are now old, and so they haven't seen hormone for several months, which is comparable to a couple of decades in a woman's life, and they're still outperforming the rats who never had estrogen in middle age.